Testing 1, 2. Oh, looks like there'll be an empty screen. Okay, I am starting. They even have YouTube Studio on. Okay. Again. I am suffering. Oh boy. Hello, everybody. I am Phylum 2. Phylum 1 is dead. Long live Phylum 2. The Phylum 1 couldn't make it because Phylum 1 is buried deep within the ground, and only a phylum of my caliber could handle the stream that is about to happen right now. Ignore the laggy fire in the background. I must let you know that this fire rages deep in my heart. and a passionate will burst forth and enlighten you on today's subject, which is the ranking of cellular organelles. Well, the old guy, you like the old guy isn't here anymore, so get used to an all new phylum. All right, ready? Oh, that is not, I did not watch this GIF for far enough because I thought it was just fire. Well, now it's fireworks, which is very fun and cool. Anyway, let's get right into it. Today, we are ranking organelles. These are cellular substructures, aka my organs, your equivalent of hearts and stomachs and all of that. We'll be ranking them in order. So without further ado, let's get into our PowerPoint. Or actually, why don't we look at the tier list first? All right, let's get this out of the way and put me up here. Great. Alrighty. So this is our organelles tier list, ranked from S to D. I have a lot of the common cellular substructures that you might encounter in your intro biology class, ranging from components of the cytoskeleton like actin and microtubules to some of, the more famous organelles. And I will be ranking all of these according to a very objective and very true criteria that I will show on screen now. There we go. Yes, this is my criteria, but before we get into it, why don't we play a little bit of music? All right, so the way that I am going to be ranking these organelles is with these three criteria. They are weighted in no particular order, but if an organelle happens to be really good at solving a problem that the cell has, I will rank it highly. Number 2 is aesthetic. It's whether or not I happen to enjoy the look of a certain organelle. And number 3 is what I like to call the POG factor. Just, are there things about this organelle that make my jaw drop, I should say. And we'll get into exactly what those POG factors are later on into the stream. But first, let's have a look again at that tier list with our first entry. This first one is called actin. Right, so actin is a part of the cell's cytoskeleton. So it is one of the components that gives cells structure. And what's unique about actin is this ability to be dynamic. So actin threads can expand and they can sort of fall apart. And this happens in a directional way. So we often call this actin treadmilling, where one side of the actin will start to grow while the other side of actin threads will start to dissolve. And so you get this really cool treadmilling effect where parts of these, you could call them bones, head in a certain direction. And when actin does that, you can get all sorts of cool things like cellular movement. So I have an illustration here. If you want to imagine what actin treadmilling actually looks like, I've loaded up something that I think many Zoomers will be pretty happy to see. See if we can see this. And if we can't, it might be a little bit of a disaster, but we'll see. Okay. Oh, looks like it's come up, but it's hiding for some reason. What's a stream without a little bit of scuff, huh? There we go. So, treadmilling can be visualized by frostwalking enchantments. So if you've ever played Minecraft and you've ever donned a pair of boots with the Frostwalker enhancement, you might be able to walk across water in a similar way to how Actin treadmills. So as you can see in this crosswalking GIF, as the player moves forward, a sheet of ice forms in front of them, and as they continue on that path, the ice that formed behind them starts melting away. So Actin is capable of doing this very thing. It grows at the beginning and disassembles towards the back. So you might be asking yourself why this action is important. Well, Actin's dynamic nature allows it to help the cell do all sorts of crazy and fun things, like move. So let's get Minecraft off the screen, and let's get this video up there. Yeah, this is macrophage movement. So macrophages are kind of my senpais. You know, they're kind of the big dogs in the immune system world, and you can see right here they are very good at moving and grooving. And that is because Actin, these treadmilling bones of the cytoskeleton, grow at these edges of these membranes, pushing them forward. So Actin is capable of doing all sorts of really cool and interesting things. So I think, when I think about Actin, I think that I would like to put Actin in... I think I have to put Actin in A tier. A for Actin. It comes up in a lot of scenarios that you wouldn't expect it to come up in. Ooh, come on, Photoshop, you can do it. There we go. Yeah, Actin, A for Actin. Yeah, A for Actin. There we go. So that looks good to me, and we can move on. I don't think that is a controversial pick, especially because when we get into Actin later on, you'll see that Actin is often involved with a whole lot more than just cells moving. So that is where I will put that. Right. Next up, we have intermediate filaments. So I decided to lump all of the cytoskeletal components together. Why am I holding anvils? I'm not entirely sure what you mean by that. Intermediate filaments. So Actin filaments are very small. They're actually nicknamed microfilaments. Intermediate filaments are a little bigger, and these proteins, which are also a part of the cytoskeleton, are incredibly diverse. So type 1 and type 2 are actually keratins, so they form the structural basis for a whole bunch of different things, like nails and hair and horns. There's a particular type of intermediate filament that I will get into later called nuclear lamins, which I will again talk about later. I always feel like I've been personally sleeping on intermediate filaments. I actually don't know a whole lot about them, but what I do know about them is kind of fun. And before you get on my case about not knowing everything about these organelles or these cellular components, need I remind you that just because you are human does not mean you know what every part of your body does or every bone in your body, right? Everybody has to go to school and everybody has to learn these things. And unfortunately, when I was out in stem cell school, I don't think I got enough of an education on intermediate filaments. So for that purpose, I think I might have to penalize them just a little bit, just because I don't know too much about them. But if there are any intermediate filament stands in the chat, you can let me know, and maybe I'll redo this tier list at a different date. So where would I put intermediate filaments given that information? Whoa, fire's back on. I think that a suitable place to put intermediate filaments would be around B tier. They've got a lot going for them, but by definition, because they are so diverse, it's kind of hard to think of them as one thing. So I think I'll give them a little more than a C, but they don't really, they're solid. They're cool if I think about them, but they don't really inspire kind of as much awe as I think watching Actin treadmill might. All right. So, next up we have microtubules. Microtubules, all right. So, microtubules are cellular railways. So we can see in this picture that the microtubules are stained in green, in that fluorescent green, and the actin is stained in that red. And I apologize if you are red-green colorblind. Researchers back then didn't seem to get the memo that might have been something to be concerned about. But yeah, microtubules are cellular railways. I think that they are pretty neat. Part of what makes them so cool is this feature called dynamic instability. Dynamic instability. Let's get that video up there. So, what we are looking at right now is a video of a microtubule forming and falling apart. This is called dynamic instability because the components of the microtubules are stable when they are bound to one another, and they actually hydrolyze in ATP, a unit of energy, to form ADP. And when that protein is bound to ADP, it is stable if it is sandwiched in between its other components. But once the tube stops building up, that top one that you see in the kind of green-blue that no longer has ATP and has become ADP, that's actually not stable. So the components in between these tubes are kept stable because they're kind of stacked on top of one another. While if the tube stops growing, then the entire thing kind of falls right apart. Might kind of remind you of what it's like to live in our post-capitalist society, building yourself up and hoping you don't fall apart. Anyway, some other cool things about microtubules. Microtubules, as I said, are little railways. They are railways for your cell to transport cargo, food, nutrition, goods, proteins, RNAs, you name it. And by railway, I really mean that almost in a literal fashion, because things actually walk on top of these railways. I'll show you that right here. Here is a video. And it's a video of a molecule called kinesin, or just rather the feet of kinesin. And if you look carefully, you can see it actually walk across the microtubule. And if you could imagine, you can imagine sticking a vesicle, some cargo of some kind, on top of those little feet. And you can imagine a very efficient way of moving things from one end of the cell to another. And I think what's fun about this video is that even though this is a cartoon, we can actually see this happen using a microscope. So this video up here, this video is a live image of cargo being transported across microtubules. So microtubules are labeled in this fluorescent red. And in green, we have labeled cargo. And that labeled cargo, you can see, it'll happen in just a second. On that right-hand side right here, you can see one of the cargoes being shipped down the microtubule. And I always think that that is such an interesting and fun thing to watch happen. So I think that if you are kind of like a train guy, then you automatically, I think, have to put microtubules pretty high. I just think that they're neat. Yeah, so let's go back to that tier list. So where would I put kinesin and dynein? Microtubules. I think because of how much I get out of watching little molecules like kinesin and dynein move across the microtubule, that it's gotta be an easy A tier. If you have ever taken a biology class, you've likely watched the Life of the Cell video where they show an animation of kinesin walking down that microtubule. You can also find a bunch of fun little meme videos of that molecule just strutting along the railway. I think because of the pog factor of watching a little guy walk down the street, it's gotta go pretty high. I think microtubules are pretty rad. All right. So microtubules are done. Next up, we have the cell membrane. So let's get to that. So the cell membrane, I think, often gets slept upon because it's just kind of like your skin, right? And we don't even really tend to think of the skin as an organ, or we don't remember oftentimes that the skin is an organ. But I'm going to make an argument for a pretty high placement for cell membranes on the tier list. Why is that? Well, I want you to imagine the early days of life, you know, way back when, before, you know, we were you were monkeys, before you were rats, all the way back to, whoa, voice crack there, all the way back to the beginning of life, this ancient and mythical time where we don't really know what happens, but we can kind of guess or surmise. So cell membranes solve a pretty important problem. Just realized I haven't used this current topic thing. I'm just gonna hide it. Cell membranes solve a pretty important problem about life, right? If we think back to the beginning when we were in the prehistoric Earth, nothing more than a bunch of chemicals floating around in Earth's oceans. Even if we know that chemistry can give rise to molecules that are biologically active, something important has to happen before we actually get life, and that is we need to make sure that enough of these chemicals are gathered in the same place. And so cell membranes give us just that thing. So cell membranes have evolved to be made out of what's known as a bilayer, a lipid bilayer, where there are two layers to your cell membrane. And these lipids, these membrane components, have two parts. In this kind of teal cyan-ish color, you can see that there are these things that look like little heads. And in between, there are these yellow-looking tails. So the gist of how these membranes come together is that these heads, these blue-colored bits, like to bind water. And these tails, these yellow things, like to bind fat. And so when you have this sort of chemistry, the tails bind the tails, and the head binds the water, which allows you to actually have layers of this fatty substance, which can wrap around on top of one another to get you protocells. So just spheres of stuff that separates junk from the outside from the good stuff that you want inside. So for problem solving, for solving a problem that life needs to solve, I think cell membranes already deserve a pretty high placement. But cell membranes are so much more than just that, right? Cell membranes do so many other things, like form tight junctions and form the structure of your organs, as well as serve as surfaces for communication between cells. And something that people, I think, forget is that cell membranes are also capable of conducting electrical current like your neurons are able to. So I just think cell membranes are so neat because there are so many things that they are actually capable of doing, and I don't think that you should sleep on them. So where would I actually put cell membranes on this tier list? pretty good question. I think that they are often slept on as something outwardly interesting, but they've got a lot going for them. But I don't think for me personally that it quite matches up with just the delight that is watching Kinesin and Dynein walk on a microtubule. So for me, I think cell membranes go in B tier. Yeah. All right. Next up, we have ribosomes. So ribosomes are little factory workers. Let's get a video of them up. Yep, there we go. So ribosomes are factory workers. So they are made of RNA, and what they do is serve as sites for protein synthesis. So in green and yellow up here, we can kind of see that those are the components of the ribosome. And these things that keep on flying in and out, these teal things that are coming, are tRNAs, transfer RNAs, that bring with them little amino acids that get stitched together in the core of the ribosome. So this line that comes in and out of the ribosome is a growing protein chain. So the ribosome is actually where that kind of work happens. So I think on a level of solving a problem for the cell, that's got to be pretty high. Proteins are the things that actually do things in the cell, right? They are the functional things that do work, basically. And so without ribosomes, you wouldn't have the proteins that you would need to actually function as a living being. So on function alone, I think the ribosome solves a brilliant problem. But there's something else, I think, that is interesting about the ribosome, right? So what I have on screen is the central dogma. That's this DNA to RNA to protein line. So information generally travels from the left to the right of this flowchart, starting at the DNA, which enables the which RNA can be read and be made from, and that messenger RNA gets made into protein, and the protein is actually the component that actually does stuff in that cell. But again, if we think about the beginning of life, you know, before we were humans or monkeys or rats, etc., 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 before when we were just a bunch of chemicals swimming in soup, it's kind of hard to imagine that, you know, a world before the first cell. The thing that makes ribosomes so compelling is that ribosomes are an example of a ribozyme, which basically indicates an RNA molecule that is able to do enzymatic work. So an RNA that can do stuff like proteins can do. Because without proteins, again, proteins being the things that do stuff, it'd be really hard to get a living organism working, right? Because you need proteins to do things, but how do you get proteins? Well, you get proteins by ribosomes making proteins. So what happens in a time before proteins? Well, it turns out that RNAs themselves are able to do things just like proteins can do. So I think this is really fun for the imagination to wonder what the world would have been like before the age of cells, right? When you think about RNAs and RNA-based enzymes being maybe some of the first biologically active molecules, I think that alone gives the ribosome a pretty high ranking. If I look at the tier list, yeah, that feeling when you're swimming in soup. I think about swimming in soup a lot. There are a lot of people who disagree on the origins of us and how we eventually came to be, whether or not we were seeded by meteors, you know, kind of like dropping matzo balls into soup, or whether or not we just spontaneously erupted from geothermal vents. The feeling of swimming in soup is an ancient and poignant one. Yeah, so ribosomes. Where do we put ribosomes on our tier list? It's a pretty good question. I'm kind of caught in between cell membrane, which I think is like a high B, and some of the cytoskeletal components. I think people that are more into thinking about prehistoric origins and engineering would probably put ribosomes pretty high. I think for now I will put ribosomes at a high B and maybe move them up a tier, depending on how the rest of this tier list goes. All right, you happy with that, chat? All right. So, now that we've done ribosomes, we are empowered and enabled to talk about an organelle that I personally have been sleeping on, which is the endoplasmic reticulum. So the ER has a fantastic name. I bet that even if it's been years since you've taken biology, there's something about the name endoplasmic reticulum that just is downright ticklish, I think. Did ribosomes always have multiple subunits? That is a great question, and I actually do not know the answer to that. So, in a future stream, I plan on doing deep dives on all of these organelles, so I can get my study on. Again, I said this earlier in the stream, but before you call me out on not knowing the answer to that question, just because you are human does not mean that you understand how every part of the human body works, and that goes the same for me. Good question, though. Yeah, endoplasmic reticulum. So, if the ribosome is the factory worker, then the endoplasmic reticulum is the factory. So, there are two parts of the endoplasmic reticulum: the rough ER, which is studded with ribosomes. The ribosomes can bind and attach themselves to the ER, and in that portion of the ER, we get a lot of concentrated protein synthesis. So if proteins are the molecules that do stuff in the cell, the rough ER is the place where a lot of those proteins actually get made. Further in the ER, you get smooth ER. This is characterized by a lack of those ribosomes, and that part of the ER does things like make different biological molecules like lipids, fats, steroids, stuff like that. So that's kind of what you would get in a biology class, right? That's kind of what you would learn. But I asked some of my fellow T and B cells, fellow immune cells, and they actually turned me on to a function of the ER that I hadn't ever heard of before. And that is the fact that the ER is a lot like creep, if you've ever played StarCraft. So what do I mean by that? So, if you've ever played StarCraft, you may know a thing or two about the Zerg race. If you don't, I did a little bit of a video as a YouTube short talking about the Zerg. But basically, the Zerg as a race can only build their buildings and structures on top of what's known as creep, which is this like biological film that expands from these things called creep tumors. So if you've played StarCraft, you're probably nodding along. If you haven't, then I'm sorry. But basically, it's this fibrous network of biological material that empowers the buildings, gives life to the buildings, supports the buildings. And the ER is kind of like that. It's big in the cell, and it makes contacts with a lot of organelles and helps those organelles do their job. And that's never something that I actually learned. That's not something that I learned early on in my stem cell days, right? This is something that I learned very recently. And those attachments to the ER actually matter a lot for those organelles, including a very popular organelle, some organelle that I think you will all be excited and will be excited to see, and that is the mitochondria. So this is really surprising. I had no idea that the endoplasmic reticulum was doing stuff like this. So it turns out, and we'll talk about the mitochondria later, don't you fret, but it turns out the mitochondria actually need help to reproduce. So cells divide, and during that division, cells need to make copies of these organs before they can split, right? You got to make sure that some cells, that when the cells split, that each cell gets roughly the same amount of stuff so that they can continue on and be a cell. And actually, the mitochondria need the help of the ER to actually do that, which is kind of mind-boggling. They kind of attach at these sites in the endoplasmic reticulum, and the ER kind of wraps around the mitochondria and helps pinch it apart. I heard about this collab. I have not heard about this collab. I was very excited to see the ER and the mitochondria collab, and I have a feeling that we might be covering more ER collabs in the future. Did somebody say powerhouse if the cell? I did not. But yeah, so endoplasmic reticulum helps the mitochondria go under fission, something I really did not expect. And for that reason, the fact that the mitochondria need the ER to split and the fact that the ER is acting in this supportive role for a lot of other organelles, I think that tickles me enough to put the ER in A tier. I think I am pretty happy with that. And I think that the collab with the mitochondria, I think, solidifies its position above everybody in B. All right. Next up, we have Golgi. All right. So there's no easy way to say this, right? I spent so long trying to figure out a way to color the Golgi as something interesting, and I just couldn't. I couldn't find it in me to find a way to make this thing sound interesting. But basically, the Golgi apparatus is the cell's post office. It gets packages from the ER, and it helps those proteins go where they need to go, and I truly cannot say anything else. If there are Golgi apparatus stands in the chat, please let me know. I don't know if you'll be able to convince me that your organelle is more interesting than watching paint dry. But please, I'll be glad to hear your thoughts, especially because by the time you get them to me, I'll probably be dead. So think about that. And with that in mind, I think Golgi body pretty easily in C tier. But I will make the caveat and say that there are no D tier organelles, just because organelles are so vital to the function of a cell that I cannot in good conscience put anything in D tier. But just know that Golgi bodies, I just think that they're the lowest of the low as of right now. The thing is, okay, the thing I want to get across, yeah, Phylum 2 is sassy. The thing that I want to get across is that I think that the Golgi is important, but that's not what this tier list is about, right? This is a tier list of how interesting I think these organelles are. And let me be clear, the post office is extremely valuable, right? We stand the post office in this chat, but am I excited about going to USPS? Not really. Not really. Am I being too harsh with lowest of the low? I don't know. But the post office, it just doesn't rustle my jimmies in any kind of fun way. That's why it gets a C tier. All right. Next up, we have the lysosome. So, the lysosome is an organelle that is very near and dear to my heart, right? The lysosome is a packet of destruction. It's basically, oh, I'm still mad. The lysosome, here, I'll be excited. The lysosome is a packet of destruction. It's the cell's digestive system, and it's got a whole bunch of cool things in it. Things in the lysosome include acids, proteases, which are enzymes that chop up protein, nucleases, which are enzymes that chop up DNA, glycosidases, which chop up sugar, lipases, which chop up fat. Basically, the lysosome is an acidic vat of biologically destructive material, and it's something that I require to do my job as a leukocyte. So us neutrophils, we are classified as phagocytes, meaning it is our job to go around and clean up garbage, and by garbage I mean bacteria. After we eat that bacteria get digested by merging with our lysosome. And so I think the process of cellular digestion is really, really cool. But where would I put this on the tier list? Now, I have to say that it is my bias to put this higher up on the tier list, right? The question is, do I want to introduce that bias onto this tier list and make it a permanent fixture of my ranking? I definitely am not going to say that it's an S tier. S tier should be reserved for organelles that are just so far and beyond the definition of cool. So I'm in between an A and a B. It doesn't really scream solves an important problem for the cell, but I mean, come on, it's got like acid and it melts things. That's just intrinsically cool, right? I think it definitely goes above intermediate filament, maybe below cell membrane. I think I'll leave it here in B tier. All right. Next up, we have the nucleus. The nucleus, as you may know, is the command center of the cell. It holds all of your genetic information. It contains what is known as the blueprint of life instructions that determine how your cells act and behave. Without the nucleus or the DNA, there would be no cell, right? And again, this is up here the central dogma. DNA, which is stored in the nucleus, gets transcribed to RNA, which gets made into proteins, which do stuff. So obviously, in terms of importance, the nucleus is pretty top tier. But there's one thing about the nucleus that kind of overlaps with the intermediate filament stuff that I talked about before, and that is the way that it solves a biological problem. So the thing that I want to talk about is nuclear reassembly. So hands up in chat if you recognize this pretty picture. This is mitosis. Yep, if there's one thing that you remember about biology, I hope that mitosis is it. The process of cellular division, right? So in this process where cells divide, something has to happen to the nucleus. It has to dissolve. So the nucleus itself has to break apart so that the chromosomes in the cell can be separated and so that there can be a clean division between the copies of your DNA. Okay. So, the question that I want to hone in on is then, how does the nucleus form again? Because by the end of cellular division, when that cell splits, you have your DNA floating in space, which presents a very interesting problem for the cell to solve. Is how do you collect all of that DNA and shove it back into the right place, right? It's essentially like if you were, I don't know, like Octomom in a Walmart or something, and you let loose all of your children to cause havoc in the Walmart aisles. How do you wrangle all of those children back together? Kind of an interesting problem. The problem worth thinking about for a little bit. How would the cell know how to get all of its floating DNA back together after the cell is fully split. And I have a bit of a hint on this next slide. See if you can figure out for yourself. You know, we're not exactly Dora the Explorer here, but I'm talking to you right now. On the screen, I have soap bubbles. I'll let you know that the way that the cell reassembles that nucleus and gathers all of the DNA back together is akin to how soap bubbles work. So it might be worth thinking a little bit about how soap works to just kind of guess how the nucleus reforms. Go on, I'll wait. Pretty interesting problem. If you can figure it out, you're better than me because I had no answer. Then again, it's not like I graduated top of my class in stem cell school. In fact, I was pretty average. Phylum 1, I think, was a lot better than me. But Phylum 1's no longer here. So the angry Phylum 2 is what you get. So how does the nucleus find all of its chromosomes, bring them back together again? It's got to do with soap bubbles. The way that the cell does this is it uses lamins. So lamina are these intermediate filaments that we talked about earlier in the stream. And these lamina form these little bubbles. So these little soap-ish bubbles really like binding to DNA. And over time, you see on the left here, those bubbles will eventually stick to those chromosomes. And when enough of those chromosomes get coated in these laminal vesicles, those vesicles start to fuse. Right? You can imagine soap bubbles, once they collide, sometimes those soap bubbles, instead of popping, will combine into like a super bubble. And eventually, those nuclear lamins will totally coat those separate pieces of DNA. And when those chromosomes are coated in that lamin, if those chromosomes happen to bump into one another, then again, it's like two soap bubbles coming together, forming one large soap bubble. So over time, it's kind of like if you rolled one of Octomom's children in sticky tape and then had that child run across all of Walmart. Eventually, that child's going to just bump into those other children, and you'll get a writhing mass of limbs. In other words, a nucleus. So I think this is super, super neat, right? If you asked me about this question, how would the nucleus reform, or how would the cell be able to do this problem of collecting the chromosomes again, I would have no idea. I would come up totally empty. But it turns out the cell and the way that the cell has evolved is a lot smarter than I am, which I think is just fun. So, where would I put the nucleus on this tier list? I think the nucleus, for the lamin problem, it really inspires a bit of fun, right? The problem of having to recollect those chromosomes, I think, really deserves an A tier, I think. It doesn't inspire quite as much childlike joy as kinesin and the microtubules does, or just the kind of creepy knowledge of the endoplasmic reticulum being like StarCraft creep. But I think it's a very neat thing for the nucleus to be able to do, is reform. All right. Last up on the list, if I could get muscle emoji react in the chat and a drum roll. You already know what's coming. We are going to talk about everybody's favorite organelle, the mitochondria. In other words, the mitochondria are the hardworking backbone of the cell. And the thing about mitochondria is that I think there are so many cool things about the mitochondria that I would not be able to cram it all in one stream. So instead, I'm just going to give you a couple tidbits. The mitochondria, we think, arose from endosymbiotic theory. In other words, a long ass time ago, before we were rats, when we were swimming in the soup of life, at some point, one of our ancestors, the cells that became the eukaryotes, ate a bacteria, a protomitochondrion. And over time, the protomitochondria and our ancestor lived together in harmony. And over time, the shell that our ancestor gave the mitochondria would protect that ancient bacteria from danger, while this bacteria that would become our mitochondria would supply our cell with energy. And what's interesting is that this strategy of cooperation was so successful that the mitochondria has no living parents left. Everything that was going to become a mitochondria already became a mitochondria, so it's actually pretty hard to find relatives of the mitochondria. You could argue that's a bit of a tragic story. But we know the mitochondria are the hardworking backbone of the cell, but what does that actually mean? Well, the mitochondria, over the gazillion years of evolution that it took to become the mitochondria, developed a novel way to extract energy from base components. And that is called aerobic respiration. So, before we talk about aerobic respiration, let's just talk about what energy production looks like in this cell. For a lot of cells, energy production is just the breakdown of sugar, aka glycolysis. Glyco coming from sugar, and lysis coming from the word for to split. And so if you don't have a mitochondria, one of the ways that you can get your energy is with this tremendously inefficient method, glycolysis. Per molecule of sugar, per molecule of glucose sugar, you only end up getting back 2 ATP. And you have to spend 2 ATP to even do it. So you spend 2 ATP, you get back 4. That means you only profit 2 ATP. Not very efficient. And it's something that almost every cell can do. It's nothing special. But you know, your ancestors, the eukaryotes, developed this relationship with mitochondria to mog the living crap out of bacteria, right? In aerobic respiration, which is what the mitochondria can do, instead of getting back 2 ATP, you get a monstrous 30 ATP for each molecule of sugar. The reason why it's called aerobic respiration is because you do, in fact, need oxygen. Making this kind of like hamon, if you are into JoJo, using the power of your breath to derive energy from sugar. Technically, if you were to write out the chemical reaction that aerobic respiration gives you, it is technically a combustion reaction. So the same kind of reaction that you would get from blowing stuff up, a combustion reaction, is happening within the walls of the mitochondria. And that's really cool, right? Technically, you are literally burning up that sugar because what you put in is some carbon and oxygen, and what you get out is carbon dioxide and water, making you little bits of, like little machines. Cool. And as opposed to glycolysis, which is pretty much accessible to any living cell that can eat glucose, that is, the aerobic respiration that the mitochondria do are only reserved for eukaryotes, cells that contain that mitochondria. So you can join the exclusive club of organisms that can efficiently derive energy from your food. The other thing that's cool about mitochondria, again, I said that they have no living parents, but that doesn't mean that they have no living relatives. mitochondria actually do have cousins. And one of those cousins is called Rickettsia, which is a bacteria that causes Rocky Mountain spotted fever. Anything that lets me think about early biogenesis, abiogenesis, early life on the planet Earth, I think automatically gets a pretty high tiering, right? The fact that we can see the mitochondria and put together this mystery of, where did we even get this thing from? And even derive the fact that, perhaps we did take in this bacteria and then lived peacefully forever after is an interesting story that is written into our history. There are so many cool things with mitochondria. The fact that they have their own DNA, right? Because they have a history of being bacteria from a gazillion years ago. And we already saw in a couple of slides earlier that they have so heavily specialized in forming this partnership with us, the eukaryotes, that they kind of lost their ability to split on their own. And they have to cooperate with our endoplasmic reticulum to get that division done. I think that even though the mitochondria is kind of the meme pick, I think we all kind of saw this coming. The mitochondria definitely deserve a spot in S tier. The thing is that is it for my tier list. What do you all think about that? Are there any organelles you would want to move up or down, or are we happy with where these lie now? I could see the argument, maybe, to bump Nucleus up to S tier, because the problem of collecting those chromosomes is a really interesting one. There's also, I think, an argument for microtubules, just because that video of kinesin gets played in every single biology class these days. But the one thing that I am sure of is that I am not moving Golgi up tier. I still think that it kind of sucks. Just kind of a mid organelle. Alright. Mitochondria, the main OP character with dead parents. That is truer than you can believe. I kind of want to think what other kind of anime tropes apply to these other organelles. Nah, none are coming to mind right now. But man, maybe that's a future short idea. I don't know. Alrighty, well, thank you. No vesicles, vesicles? That's a good question. So, yeah. So, a cellmate in chat pointed out the fact that I didn't cover vesicles. And I think that is because vesicles are kind of a broad class of things. And today I was really only prepared to talk about animal stuff. So, if I were to ever do a plant organelle tier list, vesicles would definitely be there, for sure. Microtubules S tier, yeah, I could definitely see that being the case. And also important for cell division, that is definitely true. That's an entire part of the story that I didn't even cover due to me having to get through so much. But definitely, microtubules will make an appearance at some point for a deep dive stream. Yeah, I think you can put microtubule in S tier. I think so. I think the animated nature of those microtubules is definitely worthy of an S tier placement. You've convinced me. Microtubules go in S tier. Below mitochondria though. Clathrin coated vesicle stream. True. I'd have to remember how those work, but true. I wish I could photosynthesize. I wish we could too. I actually really think that photosynthesis is super cool, but the thing is I am like super not confident in my ability to drone on and on about plants. But one day I will. In fact, I have maybe long shot hopes of collabing with a VTuber who knows about plants. I'm not going to reveal who that is, but maybe one day I will have somebody on that can teach me about all these things, because photosynthesis is literally harvesting the power of the sun, and I think that's dope as heck. Make an anime with all these guys as characters. You know what? I think that is an original idea that surely has never been done before. Dear God, I hope I make it in time to actually do this before I get scooped by cells at work again. But maybe one day. That would be fun. I don't want to go in the sun, so I don't want to photosynthesize. Good point. May I interest you in the biology of mushrooms? Do you prefer dark and damp places, and perhaps living off of the flesh of rotting meat? Who would be the one with Chunibyo syndrome? Oh, that's a good question. Which organelle is the Chunibyo organelle? No, man. Chunibyo organelle? Gosh, that's a really good question. My initial thought, and so this is like half baked, right? I haven't baked this fully, but I think Actin maybe would be the Chunibyo organelle, just because it has a very dynamic profile. It's there to support everybody, and it's trying to unlock greater powers. It's got a lot of growth, right? But maybe I'm just talking about anime protagonists. I don't know, Chunibyo organelle? So that would be an organelle that is normal, but thinks that it has special powers. I think that means it's the Golgi. I think the Golgi would then be the Chunibyo organelle, the one trying to punch above its weight. Yeah. All right. All right, cellmates, thank you for tuning into the stream. I hope you had a good time. I know that my temper has cooled since the beginning of the stream. That is because I am rapidly aging as we speak. Again, all of us phylums begin our stream at the beginning of our life, but by the end, we have aged so thoroughly that I will not see you again next time. Some iteration of me will. So, with that in mind, I would like to end today's stream by going to my works cited screen. As always, you can learn more about these organelles on your own, or just wait until I do an organelle deep dive. So go here. So here's my works cited. Here's a meme that one of the B cells that I coordinated with gave me. Golgi stands try harder. But again, if you want to cancel me, good luck. I'll be dead before you get a chance to do that. So, with that in mind, I will not see you next time, but a version of me will. So let's go and get to the end screen. Bye. Goodbye forever. I'm going to die. See you next time, except not really. Bye.